Welcome to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from neuroscientists across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of our members here. Each seminar focuses on one of the new interdisciplinary themes of Cambridge Neuroscience, which we will be launching later this year. For more info on the talks covered in this seminar series and all things neuro-related in Cambridge, please follow us on Twitter at CamNeuro and follow the links below.
Madeline is a program leader at the MRC Laboratory of Molecular Biology. So that is a unit that is located on the Cambridge Biomedical campus. And it's part of Cambridge Neuroscience and affiliated to the university. So we're really glad to have Madeline here today, and she's going to talk to us about exploring mechanisms of human brain expansion in cerebral organoids. So welcome, Madeline, and thanks for coming to talk to us.
Thank you, Darla. Thanks for the introduction, and thank you to everybody who has joined us today. So I'm going to give you a sort of a whirlwind tour of some of the recent work that my lab has been working on. And the overarching theme is brain size. And so we're really interested in understanding what are the unique mechanisms that set up our enlarged brains. Yeah, and so we're particularly interested in human-specific features and understanding what characteristics of our brains set us apart as a species, and understanding what's different during development that sets up those species-specific differences. And so, why are we interested in this? Well, actually, I mean, I think I was really surprised at these numbers. Mental health and also neurological disorders impact a very large number of people. So 1 in 4 people will experience a mental illness every single year in the UK. And those are pretty similar numbers in other Western countries. And yet mental health and neurological disorder research just doesn't receive the same amount of funding that other areas of science receive. So for example, cancer research. But it's a real area of unmet medical need. And it also suffers from something that some people have called the clinical trial cliff, which is where basically a large number of drugs have been developed in preclinical studies, like in animal models and in more sort of simple 2D in vitro cultures. But when they get into clinical trials, the success rate is incredibly poor, especially for drugs targeting the central nervous system. And the reason for that, I think, is because our brains are really quite unique. And so those preclinical models are just lacking. And so what is unique about our brains? Well, one of the most obvious features of our brains is their size. So our brains are about 3 times larger than those of our closest living relatives, the other great apes. So for example, I'm showing you here a gorilla brain. And you can see also the dramatic expansion compared with commonly used model organisms like mice. And that size is obviously determined by its cellular makeup. And so this enlarged size also means that we have a very expanded number of neurons as well, compared with, for example, other apes and mice. And so we think that this is probably one of the most obvious characteristics that we'd like to try to understand. There's, of course, other characteristics that set us apart. There are certain types of neurons that seem to be more extensively produced in the human brain. The complexity of synaptic connections is greater in humans. But just how this overall brain size is set up during development is already a really big part of what makes us human and what we're interested in understanding. So before I can sort of get into some of the stuff I'm going to show you today, I wanted to give a little bit of a background on just brain development and kind of what we know so far. So just through examination of like postmortem samples, of human embryos, and also comparing to, for example, mouse embryos, we know a lot actually about what's going on. So these kind of observational studies can give us a lot of information. So we know, for example, that the brain starts out as this neural tube, and in particular, the cortex in humans really expands out very dramatically compared with the rest of the brain. And you can see the cortex, it's filled with, it's this basically sort of thin epithelial layer early on, and then it becomes much more thickened, and then starts to generate neurons. But initially it's very thin, and it has this fluid-filled space, which are the ventricles, and the fluid inside there is the cerebrospinal fluid. And so if you were to zoom in on the developing wall of the cortex here at this stage, you'd see initially a pretty simple cellular makeup. You'd see these neuroepithelial cells dividing symmetrically and expanding. And then they switch their mode of division. They become radial glial cells because of their morphology and also their expression pattern. They start expressing glial markers. And they start to generate neurons and intermediate progenitors and outer radial glia, which are also sort of these transit sort of amplifying populations of cells that themselves can divide and then generate more neurons. And the neurons actually move outwards and create this sort of layer called the cortical plate, which will then separate sort of into these 6 major layers of neurons. And then this is then the gray matter of the brain, which is what you see then on this coronal section of a human brain. So it all starts from a really simple, actually, epithelium. And I mentioned already the CSF inside the ventricles here, and I'm going to come back to that a little bit later, but that's also an interesting component here. And so one thing we're also interested in is how the CSF is generated and how it might also impact brain development. And so because we're interested in such sort of species-specific questions, we need a species-specific model. And so that's where these cerebral organoids come in, because we can make them in theory from really any species. We just need pluripotent stem cells. That allows us the tools to now start asking questions that are not possible using more traditional in vivo studies. And so we're using, these methods were sort of first published, almost 10 years ago now. And since then, of course, we've improved things quite a bit. There's lots of new modified approaches that also other labs are using. But the principle is pretty much the same in all of them. You start with these pluripotent stem cells, you allow them to self-organize, and you essentially just provide them the right environment to develop brain tissue, just like they would in an embryo, without too much sort of extrinsic manipulation. And so what we end up with is something that looks like this. We have these large sort of lobes coming off of these. You can see that overall these organoids are very, very large compared with some of the other organoids out there. So you can see the scale bar here. So they're several millimeters in size. And each one of these lobes is also quite large. And so basically what you're looking at here, and also I should say we've injected a blue dye here so you can see these fluid-filled spaces inside of these lobes. And so basically what you're looking at here is each one of these lobes is kind of like a little cerebral cortex. And so, as a whole, the organoid it doesn't really, have the shape of a brain, but these lobes represent sort of little, cerebral units within the tissue. And so here's one where we've cut it open and sectioned it and stained for neural progenitors and neurons. And again, you can see these lobes coming off of this kind of mass of tissue. And so we're mostly just interested in looking at these lobes. And so when you compare one of these to, for example, a sectioned cortex of a mouse embryo, you start to see a lot of similarities. And when you zoom in on the wall of one of these lobes, you can also see that they're forming the layered architecture that you also see in vivo. So you have progenitors down here in this ventricular zone, and then the subventricular zone, we have this intermediate zone, and we have a cortical plate with neurons on the outside, just like the actual developing cortex. So that means that we can model all of these sort of early developmental stages that we're interested in and start looking at what might be different about the human brain. And so that's exactly what we're doing in my lab. So we're taking cells from different species, generating organoids, and just looking at them. And I should say there's some really elegant work being done in the field in this respect, particularly applying, really state-of-the-art techniques like single-cell RNA-seq and ATAC-seq, and looking at, differences in cell fate in human compared with, for example, chimpanzee organoids. But we were interested in this brain size question in particular. And we wanted to also look at, other important aspects like tissue morphology and cell biology, really. So this was work started by Sylvia Benito-Kuczynski, who was a PhD student in my lab, and she spent a while, just getting the cells, reprogramming cells, and making sure that they were making organoids with the right identity that was truly comparable, because of course we don't want to end up comparing like cerebellar organoids to cortical organoids. And so these are just some of the stainings for some of the markers that we use. So for example, in white on all of these is TBR2, which is a very specific marker to the cortex. So we know we're getting nice cortical lobes in all of these organoids and that they're comparable in terms of their identity. And what we found is that the non-human ape organoids, so the gorilla and the chimpanzee, and these are just a couple of examples, we have a couple of cell lines for every species as well. We found that they were always a little bit smaller than the human organoids, which is very interesting, of course, given the size differences that I've introduced. So then what Sylvia did was to go back and look at when that size difference first appears. And she found that it actually starts to appear really early in development of these organoids. So these are day 5 organoids, so day 5 after the neural induction. And so these are now, making those cortical lobes, and those lobes are starting to expand. And what we find is that the human organoids have larger lobes, and the lumen of these lobes are larger. So we can trace those out and then measure them and quantify them. And we can see that indeed in human organoids, we have larger lobes. What does this actually mean in terms of sort of what's going on? So if we go back to what we know about brain development, I mentioned that these cells are initially these very sort of simplified neuroepithelial cells that switch to radial glia. And this switch actually, if you think about it, can have really important implications in terms of the way the brain is expanding. So while the cells are dividing symmetrically and just expanding their numbers, the tissue is expanding in this tangential axis. So the apical surface here is expanding like this. That's the lumen. So the lumen is getting bigger, and the whole thing is getting larger in this tangential axis. Once the cells switch to making neurons, then the mode of expansion is different. Now the tissue is expanding in this apical basal axis, or this radial axis. And so you start to get thickening of the cortical wall, and the ventricles are no longer expanding. So the fact that we're seeing a difference in this sort of ventricle size in our organoids made us wonder if maybe this switch is happening later, basically, in the human context compared with the other apes, because that would enable these cells to divide and expand more and expand those lumens more before they start making neurons. So if that's the case, then we would expect neurons to start being made. If the switch has happened in the non-human ape organoids, we'd expect to see neurons. So we did some stainings, and we were really surprised to find that that is not the explanation. So we actually don't see neurons marked here by DCX, or the intermediate progenitors that also give rise to neurons, TBR2. We don't see them yet in the organoids at this early time point when we first see the difference in size, but we of course do see them later on, showing that of course they are making the switch eventually, and actually at a similar time as the human. So what's going on then here that accounts for this size difference? So what we thought about next was maybe it has to do with cell shape, actually, because So we're not seeing any difference in the cellular behaviors. They're still neuroepithelial cells. They're still just, expanding symmetrically, but could their shape be different? And could that be influencing tissue size and shape? And so we did this sparse labeling with a virus, and this is before the onset in the change, in the difference in shape and size of the tissue. And we also don't see any differences in the shape of the cells here. They have sort of typical, sort of like short, squat, fat neuroepithelial shape. But once we start to see the difference in tissue size, we also see a change in cell shape, where the non-human ape organoids are starting to show a much more constricted cell shape compared with the human. And this is also traced out here, where you can see this a little bit better. You can see that in the human, the cells are fatter than in the non-human ape. And of course, they do eventually constrict and elongate, just as they should. But it seems to be happening more slowly in human. And we can also quantify this by tracing out the apical contacts of these cells, and you can measure the size of these little honeycomb-like shapes. And when we do that over time, we can see that in both species, we're getting this constriction of cell shape, but it's happening more slowly in the human. It's taking the full entire, 7 days here to happen in human, but in the gorilla here, it's already complete by day 8. And so then we started thinking about what impact could this have on cellular behaviors. So we started to think about nuclear positioning. If cells are more constricted in their apical domain, then that could impact, where they position their nuclei. And in fact, when we quantify where the nucleus is in this apical basal axis, we find that in the non-human ape, the gorilla here, nuclei are positioned more basally. And that has important implications when it comes to another important behavior of these cells, which is called intrakinetic nuclear migration, which is where these cells move their nucleus in time with their cell cycle, so that they always undergo mitosis at the apical surface, and they undergo S phase up at the basal surface. And so we started wondering then, if nuclear positioning is different, could that also impact cell cycle? So we then teamed up with Kate McDowell, who had just started her lab here at the LMB. And she has developed this beautiful SynView light sheet imaging system, where she can image whole mouse embryos developing over time. And the beauty of the system is that we can image over several days, really, and we don't have any effect on survival in terms of phototoxicity, because these tissues and these cells are incredibly sensitive, actually, to sort of standard confocal microscopy. And so we did, again, a sparse labeling with virus, and then just watched these cells dividing, and you can see the whole tissue is also moving. So the beauty of Kate's microscope is that it's adaptive, so we can adjust the focal planes and stuff with the tissue as it's moving and changing shape. And we could do this over actually several days. So this timestamp here is actually days, hours, and minutes. And then we could just trace the cells and count how long does it take to go from one mitosis to the next. So how long is the cell cycle? And what we found is that during this phase, when cell shape has changed already in the non-human ape context, but it hasn't yet changed in the human, we find that cell cycle is shorter in these gorilla organoids. And it's not that much shorter, it's only about 3 hours shorter, but it has important implications in terms of the numbers of cells that are generated in that period of time. So you can do a bit of just simple sort of growth curve modeling and calculate how many cells would that enable these cells to generate in that amount of time, given this difference in cell cycle length. And what you find is that the prediction is that this would lead to around almost a 2-fold increase in the numbers of progenitors and then the number of neurons that are being generated in human compared with the non-human context. And I think that's really interesting, because I told you at the beginning that brain size is around 3-fold larger in humans. But when you actually count the number of neurons in the cortex, you find that our cortex has about 2-fold more neurons than the gorilla or the chimpanzee. And so this actually helps to account for a large amount of that. And so I think it's really shedding light on what might really be setting up this great increase in overall brain size in humans. And so I'll just like wrap up this part and just tell you about what we found. So we found these sort of founder neuroepithelial cells seem to be making this transition in cell shape. That in and of itself is really interesting. It hadn't really been described before that these cells undergo a shape change first before they change their fate and their identity. And so that seems to be sort of a precursor to this whole transition. And so we've called this the transitioning neuroepithelial stage. And then because this is happening a little bit later in humans, that's having an effect on their cell cycle length during this time. And that's leading to an increased number of progenitors, so that once they start making neurons, there's just more founder stem cells that then can generate more neurons. And so we think that's a potential explanation for why we have such enlarged brains. And it really fits actually with the comparative neuroanatomy, because actually when you compare a human and a chimpanzee brain, what you find is that neurons are just generally increased. All neurons are generally increased in humans compared with chimpanzees. And so it would suggest that something has to happen differently in the human before any neurons are made, so that it can equally affect all of the neurons that are subsequently generated. And that's exactly what you see when you compare a human and a chimpanzee brain. So now I want to switch to the cerebrospinal fluid, and in particular the choroid plexus, which is the tissue inside of this ventricle that's actually generating the CSF and pumping it into these ventricles. And the choroid plexus is a very important tissue. A lot of you may have not even heard of it, but it's a very important part of the barrier that protects the brain. So it filters blood and then generates CSF from that blood, but it has to make sure, of course, that only what's needed in the brain actually gets into the CSF, because once something is in the CSF, it has free access to the brain. And in fact, that's why some people actually give drugs that way, intrathecally, into the spinal cord, into the spinal column, because once you can get a drug into the CSF, it's got access to the brain. So this choroid plexus, which is generating CSF, has to be just as selective as the more widely studied blood-brain barrier, but it's part of these broader sort of brain barriers. And some of you might have even seen some very recent work that made a lot of headlines, suggesting that cerebrospinal fluid actually is very powerful fluid and can actually improve memory in old mice. When you transplant young CSF from young mice, transplant it into, basically infuse it into the brains of older mice, you're able to improve their memory. So we're very interested in the CSF that's being produced in this developmental context, and what might be present in it that might also then influence the developing brain. And so to look at that, this was work carried out by Laura Pellegrini, who's a star postdoc in the lab and has just set all of this up beautifully and has been able to actually generate choroid plexus organoids that make CSF. And so this is the protocol that she developed. It's based on our standard sort of cortical organoid, cerebral organoid protocol, and it simply involves a little bit of patterning a bit later on to help dorsalize the cerebral tissue so that we get choroid plexus, which is really in the dorsalmost region of the developing cerebral cortex. And so simply by having this BMP4 and cheer pulse at just the right time and the right amount, we're able to generate these beautiful organoids that have a structure that very much looks like the real thing. They have the right polarity, so you can stain them with markers like aquaporin and TTR, and you can see that they have the proper sort of apicobasal polarity. They have all of the characteristics of a highly secretory epithelium. They have tight junctions and microvilli, extracellular vesicles, and multivesicular bodies. And in fact, actually, as they mature, they start to make these really large fluid-filled sort of bubbles or sacs coming off of these tissues. And you can actually see as well that the fluid inside is not simply media, because here's one where we've actually left it out of the incubator for a while, so the media got really pink. You can see that the fluid inside is clear. And so that suggests that there's a barrier forming there, that it's not just media leaking in. And in fact, if we stain for some of these markers of tight junctions, these claudins, we can see them nicely expressed and expressed in the right apicobasal polarity. And we can also put on different things. So in this case, we've put on different sized dextrans that are fluorescently labeled, and we can see that when we put them in the media, they aren't really making their way into the organoid fluid. And so we've also tested this more specifically by looking at selectivity to small molecules. And this is something that, people have been trying to develop blood-brain barrier models in vitro for a long time. And there's some success there, but they're always a little bit more leaky than the real thing, actually. And so we really wanted to really push the limits of the system, actually. So we decided to go for 2 small molecules that are very, very similar, very small molecules, very similar, but show very different behaviors in terms of being able to cross into the brain. And so we chose L-DOPA and dopamine. So it's long been known that dopamine doesn't really cross into the brain. And that's why we give L-DOPA, which is a precursor to dopamine, to patients with Parkinson's disease, for example, because it does cross into the brain. And so what we did here was to just put these 2 molecules into the media of our organoids, collect fluid, and then do NMR and measure whether they were present in this fluid inside the organoids. And we could see very nicely that L-DOPA was able to enter the organoid fluid, but not dopamine. And this is also quantified here. We then looked at a broader range of different molecules. Here's a couple more. Here's bupropionyl, which is known to cross into the brain. Methotrexate does not. And just like in vivo, we see it bupropionyl crossing in and methotrexate not. And so we just looked at a couple of these different molecules and then compared them with in vivo measurements. And importantly, this is measurements from human. And that's really important because actually the kinetics of these drugs is really different from species to species. And so we wanted to make sure we compared with human. And when we do this, we see a very nice correlation of the amount that's getting into the CSF-like fluid inside these organoids versus the media. But I think what I was really struck by is actually the slope here that is almost exactly 1, which means it's not just a great correlation, it's actually a 1-to-1 readout. So it's actually quantitatively predictive of how much of the drug will actually enter into the CSF if it's in the blood. And so we also decided then to look at some sort of unknown drugs. So in this case, we turned to one called BIA 102474. This is a drug that was actually tested in a phase 1 clinical trial, and this is just the safety testing. And unfortunately, actually killed 1 person, and it left 5 more with brain damage. And that was completely unpredicted in the preclinical work. And what was kind of interesting about it is that the patients that experienced these severe side effects were the ones who had the escalating dose. So the dose was being increased over time. But interestingly, the symptoms appeared and lasted even after there was no more drug in the blood. And it took a while as well for these symptoms to appear. So there were some suggestions that maybe the drug was actually accumulating in the brain. And this was not predicted in any of the animal models. So clearly something species specific going on here. So we thought maybe our organoids would be a good model to look at this and see whether they could have predicted this. And what we found was that just like bupropionyl, it seems to be able to enter the organoid very nicely. It was, of course, known. And in fact, it was a drug they were trying to use to treat a neurological condition. So the goal was to get it into the brain. But what I think they couldn't have predicted was what we are also seeing in the organoids, which is an accumulation. So we're basically, the drug isn't actually leaving the organoid like it does, like bupropionyl, for example. And so I think, I think maybe, if we'd had some of this data, we might have been able to predict that this drug might be accumulating and we shouldn't be doing an escalating dose of the drug, and we could have adjusted the dosage as well. And so I'm hoping that this will be informative for future studies and hopefully future toxicity. And also in terms of sort of predicting how much, because often with these phase 1 clinical trials, you really go in blindly. You sort of take what you have from the animals, but the actual dosage can be very different. Like, for example, L-DOPA, you have to give a completely different dosage to humans than you give to mice to get the same amount into the brain. And so there's clearly species-specific differences there. And so having a quantitative predictive model that we can use to start more intelligently designing these trials and choosing the dosage, I think would be really powerful. So now going back to the fluid, we of course got very excited as well about the fluid and the fact that it's, you know, this clear fluid. It looks like CSF. We wanted to see, is it CSF? So what we did is extract fluid, send it for mass spec, and then compare with some in vivo CSF samples that we got. And what we found was really great overlap in terms of the protein content that we're detecting in these organoids, in this organoid fluid and in vivo CSF. And really nicely, we can also see a number of markers here, you know, putative biomarkers that people are starting to use in the clinic. And so I think this also has some applicability in that front as well. So I'm going to end that part there and now switch gears again and talk about another project that we've just recently published. And this is a project looking at sex-related brain differences. And this is work that was totally spearheaded and run by a very talented postdoc, Eva Kalava. And what she became interested in when she joined my lab is this difference in susceptibility to different neurological and mental health conditions in males versus females. And so, for example, ADHD and autism spectrum disorder affect males much more than they do females. And we really don't understand why that is. And of course, there's a lot of questions about diagnosis, but I think there is also something interesting going on in terms of the actual biology. And so we wanted to look and see whether we could model some of that. And, you know, it suggests that could there already be something a little bit different about the male and the female brain that might be setting them up for, you know, for differences in the susceptibility. And, you know, this area of research is kind of controversial, and there's some data out there that's not necessarily very, you know, well powered. But one thing that I think is very clear, and it does come up time and time again, is a difference in brain size. So when you look across an entire population, you do find that males have on average a larger brain size than females. Of course, males also have larger bodies, so that kind of goes hand in hand. But it's an interesting question, you know, how does the male brain end up getting a little bit larger? What is telling it to actually grow? And I should also state, of course, that these differences are on a population level, and on an individual level, you really can't tell because there's really a lot of overlap. But it does suggest that maybe there's something already, you know, different going on that might be, that might even happen during development. And in fact, when you look at differences in gene expression in the brain, you find that the biggest differences are present during fetal development compared with, you know, postnatal or adulthood. So again, because brain size is also something that's being set up during development, we thought that maybe the organoids could be a good model to look at. And so there are basically two possible factors that could lead to sex-based brain size differences. One is hormones and one is genetics. So, you know, XX or XY, and hormones, you know, testosterone and estrogen. And actually, it's actually been shown that during development, the male fetus in particular has a very large, you know, there's a very high exposure to testosterone actually. So the fetal testes, as they're developing, actually start producing really large amounts of testosterone, almost the same level as you see during the pubertal surge, which is really high. So what effect could this testosterone be having on the developing brain? Could that be involved in some of these sex-based brain differences? And how much is it really just because of the circulating testosterone versus, you know, the XX or XY chromosomal complement? And of course, in vivo, it's pretty much impossible to separate those two, because obviously an XY fetus is going to have testes and is going to be producing testosterone. So it's very hard to disentangle those two possible contributors. But with organoids, we can do that, because obviously we have them outside the body. We can control how much hormones they get, and we can make them from male and female lines. And so that's exactly what Eva did. And so she took these organoids from male and female lines and she exposed them to different hormones. So T is testosterone, E is estrogen. And DHT is a form of testosterone that can't be converted to estrogen, because actually testosterone and estrogen are very closely related. They're very similar molecules. And there's one enzyme that can convert testosterone into estrogen, and that's aromatase. And actually in mice, we know that masculinization of the brain comes about because of aromatization of testosterone to estrogen. So counterintuitively, in the mouse context, estrogen is the masculinizing hormone. So by putting on DHT, which can't be converted to estrogen, we're also testing whether that's true in the human context as well. So basically, we would predict that if it was like in mice, then we should see the same phenotype with testosterone and estrogen, and DHT wouldn't have that effect. But if it's different, then maybe DHT and testosterone would actually show the same phenotype. So if it's the androgens themselves, then we should see the same thing with the DHT and the testosterone. And so basically, Eva then looked at a number of different behaviors during neurogenesis, cellular behaviors, I mean. So she looked at these stem cells, she looked at these intermediate populations, these intermediate progenitors and outer radial glia, and she looked at the neurons. And so I'm just going to show you some of the phenotypes that we saw, but basically it's all pointing to an effect of androgens on progenitors. So for example, if we look at intermediate progenitors, which are marked here by TBR2, we find that in both DHT and T-treated organoids, we can see an increase in these intermediate progenitors. And we don't see that with estrogen. We see the same thing when we look at both XX and XY organoids. So we don't see any differences between the XX and XY. And we see that both of them respond to the two androgens the same and increase their intermediate progenitor numbers. When we look at where this increase in intermediate progenitors is coming from, using a lineage tracing approach with a virus. So we're labeling the stem cells that are going to give rise to those intermediate progenitors. We find that when you treat with the androgen here, this is the DHT, we can see an increased size of clones. So this is basically the stem cells are more proliferative. So they're generating a larger clone size when you treat with the androgen. And another way of measuring this is by overexpressing a constitutively active androgen receptor and then looking at proliferation using this proliferation marker Ki67. And again, we see that when cells are expressing this androgen, this constitutively active androgen receptor, we see an increase in their proliferation. We then did RNA-seq to look at the mechanism of this and try to find out what was happening downstream. And we found a number of differentially expressed genes. And when we do go-term analysis, we pull out a lot of things related to chromatin and HDAC activity. And that's really interesting because actually going back to what I said about differential, susceptibility to different conditions, things like autism disorder and actually like also schizophrenia, for example, there's been a lot of recent work showing that epigenetic factors seem to be involved. A number of mutations seem to affect different factors that are involved in epigenetics. And HDAC in particular actually has been involved here. So we were really interested in this HDAC activity and these two HDACs. So we also decided to look at that by using this HDAC inhibitor, VPA, valproic acid. And when we do that, we actually see that it does the opposite of the DHT and that we can rescue that with the DHT, suggesting that the two are acting together to impact progenitor proliferation. We also, if we look, instead of Go terms, now we look at human phenotype ontology. We pull out a lot of terms related to brain size, microcephaly, macrocephaly. And that makes sense when we think about what I just said in the beginning about brain size differences. And so then what Eva decided to do was to actually look at neuron number and try to understand, so obviously brain size is going to come back to cellular makeup. So we would expect increased numbers of neurons. And so when we look at these, this marker SATB2 of excitatory upper layer neurons, we can see that these are increased in the androgen treated organoids. Now, very interestingly, if we instead look at the progenitors that are going to give rise to inhibitory interneurons, we don't see a difference. So these are marked by DLX2. This is kind of similar to like TBR2, only it's for the region that generates inhibitory neurons. And here we don't see any differences. If anything, we see a slight decrease. So it seems like androgens are specifically acting on excitatory neurogenic progenitors and increasing their proliferation, and that's then increasing their neurogenic output. So we're getting a specific increase in excitatory neurogenesis, but not inhibitory, which is really interesting. Again, going back to autism spectrum disorder, for example, where it has been proposed that there might be something going on with the excitatory inhibitory balance, that may be imbalanced. And so I think what we're finding is, it's been long proposed that there's some sort of like female protective effect. And it's been recently shown that females with autism spectrum disorder have more genetic mutations. They have like a higher mutational load in order to show the phenotype. Why would they need a higher mutational load? I think the idea here is that maybe males are already sort of predisposed to some extent. And then on top of that, you add to it a mutation or two and you tip the balance too far. Whereas females are not so predisposed. So you would need more mutations in order to tip the balance, in order to tip this excitatory inhibitory balance. And so, we're hoping that this starts to shed light on why there's differences there. And I think it's also really interesting in terms of thinking about, environmental aspects as well. So for example, could there be androgenic compounds that in the environment that might be impacting the developing brain, and maybe we should start investigating some of those as well. Right, so now I'll just thank everybody. So I think their pictures came up throughout. I also want to make sure I highlight Magda Sutcliffe because she contributed to all of the projects I showed you. She's our senior research technician that helps with all of the projects really. And I want to make sure I thank my collaborators and my funding. Thanks.
Thank you, Madeline, for that wonderful talk. Join us next time when we welcome Professor James Rowe, a professor in the Department of Clinical Neurosciences and the MRC Cognition and Brain Sciences Unit. He is the director of the Centre for Frontotemporal Dementia. James studies the mechanisms and treatment of cognitive disorders, including frontotemporal dementia, progressive supranuclear palsy, and Alzheimer's disease. He'll be speaking to us on apathy and impulsivity in neurological disease, cause and effect and treatment. Don't miss it. For more info on the talks covered in this seminar series and all things neuro-related here in Cambridge, please follow us on Twitter @CamNeuro and follow the links below. See you next time.